Welcome, everybody, to Science Update. This is a Facebook Live session where I take on your science faith questions. This week's question comes from an unusual source. It's from a Facebook group that I'm a member of called Old Earth Creationists. And while I don't spend a lot of time on that Facebook group, from time to time I am notified of conversations that are happening on that group. And this happened last week. When I was alerted to a discussion taking place based on an article that somebody posted to the group entitled, An Ancient Virus May Be Responsible for Human Consciousness. And through back channels, I was asked to comment on this particular article, which was published on the Live Science website, I think February 2nd, 2018. And so, given that request, I thought this would make an excellent Facebook Live topic. And this particular news item that was published on the Live Science website is based on a couple of papers published in the journal Cell towards the end of January. And two separate research teams basically argued independently towards the same conclusion that it looks as if evolution employed an endogenous retrovirus, which would be a particular type of junk DNA that's believed to be derived from a retroviral infection, that evolutionary mechanisms basically co-opted and modified the genes from an endogenous retrovirus to create a biochemical system that will help to strengthen the synaptic connections between neurons. And many people believe that this process is responsible for synaptic plasticity, which allows for things like learning, conditioning, cognition, and even self-awareness to take place, with the implication being that retroviruses and the remnants of retroviral infections in the genomes of animals has played a key role in essentially shaping and evolving the brains of animals to the point that human beings have not only the capacity for advanced cognition, but also the capacity for self-awareness. So I'm going to go ahead and unpackage this particular set of discoveries and this Live Science news article in just a minute, but as is customary, I would ask for those of you who are watching this broadcast to please take a moment and check in. Let me know who you are and where you're watching from. This is valuable information for me to have as I plan future broadcasts, but also it's a great way for me just to get to know who you are and who's watching these broadcasts. And that includes, with last week's broadcast, Colleen Reith from Houston, Texas, Don Snell from Portersville, California, Paul Frank from Cheney, Kansas, and then Paul Peterson from Hudson, Wisconsin. And I see that Rick Viduka has just checked in and Dave Steensland has just checked in as well. So welcome to all of you who are checking in to watch this live, but also thanks to all of you who have watched the video posts of this broadcast and have taken the moment to check in and say hi and let me know who you are. Also, if you don't mind, react to the broadcast with the like button. The more reactions that we get to the broadcast, the greater the visibility this broadcast has in the Facebook feed. So by liking the broadcast or offering comments on how the broadcast could be improved, you are actually helping to create greater visibility for the broadcast. So thank you so much for doing that. And then last but not least, I really want to know what you think about this particular question. Is it possible that evolutionary mechanisms could take the leftover remnants of retroviral infections and co-opt those genes or those DNA sequences more appropriately and use them to essentially shape the history of animal evolution, including the generation of brains and the evolutionary advance and progression of brain structure and brain capacity? Or do you think that the history of life on Earth is better explained through the handiwork of a creator? I want to know what you think. even if your perspective differs from mine. Okay, let's go ahead and get started by taking a look at this news item. And what I'm going to do is focus my attention more on the papers published in the journal Cell that fueled this particular article. So I'm not going to respond to the article point by point, but rather focus on the work that was published in the journal Cell. And I am going to just simply present the work as it was published, meaning that it was essentially presented in the context of the evolutionary paradigm. And then after I do that, I'll then begin to lay out how I think about this work by these scientists that again published their work in Cell from an old Earth creationist perspective, from an intelligent design / creation model perspective. Now, before I get into the work that was published in Cell, just a little bit of background information I think is necessary to help you appreciate the context of this work, which is, by the way, a very fine piece of biochemistry that these two independent research teams did. But one of the big questions in neuroscience today is, how does neuroplasticity work? And what I mean by that is the idea that as information is processed by the brain, as information is stored in the form of short-term and long-term memories, there's plasticity that takes place in the connections between different nerve cells in the brain. And that point of connection is referred to as the synapse, which is a gap or a space between adjacent nerve cells. And the idea here is that by strengthening these synaptic connections in some instances, or by weakening synaptic connections in other instances, a network of neurons is created that essentially, again, serves as the framework to process and store information in the brain. And people believe that this strengthening and weakening of synaptic connections plays a role in conditioning, in learning. It plays a role in memory storage. It plays a role in cognition, and even our capacity for self-awareness. And so again, neuroscientists are trying to understand what is happening here at a molecular level, at a molecular scale that's responsible for strengthening these synaptic connections. Now, neuroscientists believe that what is going on to strengthen a synaptic connection is the exchange of chemicals between two nerve cells, where those chemicals are secreted by one nerve cell and then traverse the synaptic space and then are taken up by another nerve cell. But nobody quite knows exactly how this mechanism works, or exactly what's happening once the chemicals are taken up by nerve cells. And so the research teams that were looking at this question as separate independent research teams essentially came to the same simultaneous conclusion about a facet or an aspect of how this synaptic strengthening takes place. And they were focusing on a particular protein called the ARC1 protein, which of course is encoded by the ARC1 gene. And for those of you that are keeping score at home, the ARC acronym stands for Activity Regulated Cytoskeleton Associated Protein. And it's interesting because this particular protein has been implicated in a number of neurological disorders: Alzheimer's disease, schizophrenia, and even neurodevelopmental disorders, where mutations in the ARC1 gene seem to have, again, an association with these kinds of disorders that seem to involve a loss of cognition to some capacity. Also, there are other studies that have identified, again, a role that ARC1 plays in synaptic plasticity. And also, people have noted that when the ARC1 gene is transcribed to produce messenger RNA, that messenger RNA migrates to what are called dendrites in the nerve cell. In a nerve cell, you have the cell body, and then there's a long extension from the cell body called an axon. And then there's also these tree-like extensions, processes that extend from the cell body that are referred to as dendrites because they have kind of like a tree-like shape. And it's the dendrites that make connections with axons of adjacent nerve cells or with other dendrites from adjacent nerve cells. And these interactions are what form the synaptic connection between nerve cells. So it looks as if that messenger RNA, once it's transcribed, makes its way to the dendrites, and it looks like that's where it's translated to produce the ARC1 protein. Now, something that's very interesting is that when people have examined the genes, the DNA sequence for the ARC1 gene or the amino acid sequence for the ARC1 protein, they've discovered that it bears a striking similarity to something called the GAG protein, the GAG protein, which stands for Group Antigen Specific Protein. This is a protein that is associated with retroviruses. Now, a little bit of an aside for those of you that are not familiar with what viruses are. Viruses are these infectious agents that consist of proteins and nucleic acids. The viral genetic material can either be single-stranded or double-stranded RNA, or it can be single-stranded or double-stranded DNA. And that genetic material is encapsulated in a protein shell that's formed from a number of individual proteins that aggregate to form this hollow capsid or this hollow shell. And sometimes viruses will even be surrounded by a plasma membrane type structure. And a virus will interact with the cell surface, and when it does, it either injects its genetic material into the cell or the entire virus is taken into the cell through a variety of different mechanisms, where ultimately the genetic material associated with the virus is introduced into the cytoplasm of the host cell. And that genetic material will then take over the operation of the cell's machinery to make more copies of the genetic material, but also to transcribe genes that are part of the viral genome that typically will produce proteins that will form the viral capsid. And so once that happens, that viral capsid will assemble typically around the viral genetic material. And then there is a process by which that virus will interact with the plasma membrane, and typically an extracellular vesicle will, derived from the plasma membrane of the host, will surround the viral particle, and it will be released into the extracellular space, and in doing so, now can allow the virus to repeat that infectious cycle. Now, retroviruses are a special type of virus that have RNA as their genetic material. And so when that RNA enters the cell, it is reverse transcribed by an enzyme named reverse transcriptase that converts the RNA into DNA, and then there are enzymes called integrases that will incorporate that retroviral DNA into the genome of the host cell. This is what happens, for example, with the HIV virus. And when that happens, that retrovirus can remain active or dormant in the cell once its genetic material is incorporated into the genetic material of the host cell. But occasionally, it's believed that these retroviruses that have become incorporated into the host genome will undergo severe enough sets of mutations that it renders them non-functional, and evolutionary biologists believe that this is the source of what are called endogenous retroviruses. But because of the similarity between the GAG protein of retroviruses and the ARC1 protein, these two research teams independently speculated that maybe the mechanism for transmitting information from one neuron to the other is going to be retroviral-like, where the ARC protein would form a capsid that would surround its own messenger RNA and form an extracellular vesicle that would transverse the synaptic space and be taken up by adjacent neurons. And so, in order to test this idea, these two separate research teams focused on two separate model systems. One team looked at Drosophila, the fruit fly, and the other looked at a mouse model. And for the team that studied Drosophila, they noted that the ARC1 protein would actually form capsid-like structures. When they purified the ARC1 protein in a laboratory, they could get it to form these capsid-like structures. That these capsid proteins would bind messenger RNA in the 3' untranslated region, for those of you that are keeping score at home. That they could actually, once associated with the messenger RNA, form extracellular vesicles that would essentially be transferred from motor neurons to muscle cells in Drosophila, and that if they blocked the activity, they could basically disrupt synaptic plasticity in Drosophila. The team that studied the mouse model observed almost identical things. They saw that the ARC1 would assemble into capsids that would encapsulate RNA that could be released from neurons into the synaptic space as extracellular vesicles, that these vesicles could transfer the ARC1 messenger RNA from one neuron to the other neuron, where it would take up residence in the cytoplasm of the neuron that was receiving that chemical signal. And then finally, purified ARC1 capsids could transfer ARC1 messenger RNA into neurons. They could use it to deliver messenger RNA into neurons. And so because of the similarity in these mechanisms, the researchers argued that it looks as if essentially a retroviral infection that then, in which this retrovirus became endogenized, was the raw material that evolution used to create a retroviral-like process of infection that instead was now being deployed by neurons to, again, build up the strength of synaptic connections, and that this happened very early in animal evolution, very early in the origin of land vertebrates, very early on in the origin of insects, and that because of that, that this mechanism was critical for, again, shaping the evolution of mammalian brains and the evolution of insect brains. Now, something else that's also interesting is that they note that it looks as if the ARC1 sequence, gene sequence, was actually derived from a retrotransposon family, which would be a family of DNA elements that would be derived from a retroviral infection where that retrovirus has become endogenized. And particularly the family is the TY3 / gypsy retrotransposon family. So when you put this all together, it looks as if this is really a very compelling case for an evolutionary origin of mammalian brains and insect brains, where retroviral infections seem to be playing a key role. In fact, the researchers point out that probably 50% of the mammalian genome seems to be derived from retroviral infections, and they argue that this means that retroviruses must be a key driver of evolutionary history, and that this is just one example of that. Now, I would agree with people who would embrace the evolutionary paradigm that this seems to be like a very compelling story for, again, an evolutionary history for animal life on Earth, and a very compelling argument that key brain activities are ultimately produced through this evolutionary mechanism of co-option and then neo-functionalization of so-called junk DNA that, again, arises from retroviral infections. So how then do I make sense of this from the standpoint of an old Earth creationist? Is it possible to understand this data from an intelligent design / creation model perspective? Well, I'm going to answer yes to that question. In fact, I'm going to go one step further and argue that if you take a ID / creation model perspective, it makes better sense of this data than does an evolutionary explanation. Now, to get to that conclusion, I need to unpack a few things. First of all, we need to take a breather from talking about the science and talk a little bit about philosophy, because here the philosophy that undergirds science now becomes very, very important. For I think many of you are aware of this, the philosophical framework for modern day science is methodological naturalism, that basically says in a nutshell this, that whenever we approach trying to understand some kind of phenomena in nature, we have to employ materialistic explanations that evoke mechanism, that ultimately mechanism alone must be the way that we explain the features that we observe in nature. That is, we cannot appeal to the work of an intelligent designer or the work of a creator. Mechanism alone has to be the way in which we understand phenomena in nature, including, for example, the origin and the design of this very elegant mechanism for basically synaptic strengthening that involves this retroviral-like mechanism. Now, this approach to science is, in my view, highly restrictive. I wouldn't argue against methodological naturalism as such, because it's a very powerful framework that ensures that we avoid God-of-the-gap explanations, that we really focus on trying to understand how things happen. But when it comes to why things happen, methodological naturalism really falls short. And I do believe there's instances where methodological naturalism breaks down, where we do see evidence that does indeed look like an intelligent agent must have been involved. But because of the restrictions of methodological naturalism, we can't let the evidence lead where it should naturally lead us. That is, science no longer becomes an enterprise where we're looking to discover truth, but looking to discover naturalistic, mechanistic explanations. And so the long and the short of that is that if that's the approach you're going to take to understanding, let's say, the design and the history of life, then you have no choice but to say, ultimately, evolutionary mechanisms must indeed explain the origin, the history, and the design of life, because you have no other options available to you. So even if those explanations fall short, you still have to insist that somehow evolution must be the way to explain some feature that we see in biology, because again of the influence of methodological naturalism. It's not bad. It's not a bad philosophy for science. It's just a limited philosophy of science that indeed does break down, and I think sometimes can prevent us from understanding and discovering the truth about the why nature looks the way that it looks. But what if we relaxed, hypothetically, this requirement of methodological naturalism and said we're willing to allow for design or creation model explanations for phenomena that we see in biology? Is it possible to make sense of this set of discoveries in that framework? Well, I'm going to answer yes to that question. And it's interesting because when we look at the history of biology, we note that prior to Darwin, biology was teleological in nature, where people were looking at both mechanism and design as ways to account for features in biological systems. The work of William Paley in the late 1700s, Natural Theology, where he argued that the design that we see in biology reflects the handiwork of a mind, influenced the way biologists thought. And there was a debate going on among biologists as to whether it was function or whether it was form that were the overarching ideas to interpret biological design, where you had people like Richard Owen, who was arguing that form was as important as function, and was actually looking to unite those two schools of thought with his theoretical work on the vertebrate archetype. But Owen argued that shared biological features that are observed among organisms that would naturally group together reflect an archetypical design that exists in the mind of a creator that is then functionally manifested in the created order. So for Owen, if he saw observations like we're seeing now, where we would say, well, there's a similarity between the retroviral infection mechanism and the mode by which this ARC1 messenger RNA is transmitted from neuron to neuron, Owen would say, well, this reflects an archetypical design that exists in the mind of a creator, as opposed to saying this homology reflects an evolutionary history. So Owen saw this kind of data as reflecting, again, the work of a mind, and that he was comfortable thinking about biological designs in a teleological framework, in a design-friendly framework. But Darwin comes along and he proposes his theory of evolution to explain the origin of species, and then argues that that mechanism could be extrapolated to explain not only life's major groups, but the design that we see in biology, where now Darwin argued that homologies reflect a common descent from a shared ancestor, where that descent with modification takes place through the guidance of this blind process of natural selection. What Darwin did, which was so appealing to biologists of his time, was he made biology mechanistic. He took it from a teleological enterprise to a mechanistic enterprise. And in doing so, moved biology into the same category as all the other sciences. Prior to Darwin, biology was the last of the sciences that was teleological in orientation. Physics and chemistry were centered around mechanism. So was astronomy, so was geology. And so what Darwin did that was so appealing was he introduced mechanism into biology and argued that that mechanism could explain everything. We no longer need a creator. And so what Darwin did was essentially dovetail his idea of mechanism being the forefront of biology with this emerging concept of methodological naturalism. And as a result of that, that shapes the way that people now view biology, is from a methodological naturalistic framework where mechanism, namely evolutionary mechanisms, are how we account for features in biology. Now, I know that I've belabored that point, but what I'm trying to illustrate here is that there's a precedence, a historical precedence to look at shared features as reflecting an archetypical design as opposed to the outworkings of evolutionary mechanisms. And so you could argue again that the shared similarity between the retroviral infectious process and again how this information transfer takes place between neurons could again be understood from a common design framework. So the question is now, with those two models on the table, which is a better explanation? Well, I'm going to argue in the next few minutes that I think the best explanation is one that employs common design, not common descent. So, for example, one of the things that's very interesting here is that the central role that this mechanism plays in essentially brain function. So it's one thing to say we've got a piece of retroviral DNA that's become endogenized and that maybe we could co-opt part of that to generate a new protein or to generate a new regulatory element that will influence the expression of a single gene. But it's another thing to say that that mechanism of co-option would create a complex, would be central to creating a complex mechanism that is sophisticated, that is elegant, that is mind-boggling in it, not only in its elegance and sophistication, but in its complexity, that would then assume a central role in cell physiology and in the higher physiology of the organism. That to me is intuitively seems to be really unrealistic to say that something like that would happen. But what to me is the clincher is the second point of concern for me, and that is that these researchers discovered that this ARC1 mechanism in Drosophila and in mice must have evolved independently two separate times. That Drosophila represent one branch of the evolutionary tree of life that are referred to as protostomes, whereas the other branch are referred to as deuterostomes. And it looks as if this mechanism for synaptic transmission and this mechanism for synaptic plasticity evolved two separate times independently in insects and in mammals, and yet the mechanism is identical in both cases. That to me is mind-boggling to make that particular claim, and to draw the conclusion that evolution could create such an intricate, sophisticated system that is so central, again, to cell physiology and higher physiology in the organism, that to me is absolutely an astounding claim. And the reason why I say that is because when you look at the nature of the evolutionary process, it is historically contingent. This is the term that Stephen Jay Gould coined, where he basically argued that that evolution is predicated on a sequence of chance events. And if you alter any one of those events, it's going to send evolution down a very different trajectory. And that if you rewind the tape of life and replay it, the outcome is going to be different every time because of, again, the historically contingent nature and the sequential nature of evolutionary transformations. And Gould argued that evolution should not repeat itself, or if it does, it should be an extremely rare occurrence. And yet, it looks as if evolution has indeed repeated itself in this particular case. Now, I know that there are some of you out there that think that this idea of evolution being non-repeatable isn't something that flows out of the evolutionary paradigm. I would disagree because, again, Gould made that very argument. But since Gould's time, there have been a number of experiments where people have monitored, let's say, the long-term evolution of E. coli in a laboratory where they show that microevolutionary changes in E. coli are historically contingent. There are other people who have been studying protein evolution, arguing that if evolution could explain the genesis of proteins, that it happens in a historically contingent manner. That is, that these are observations that are continually being made by evolutionary biologists, that it looks as if evolution is historically contingent. If that's the case, we would not expect independent origin events, and that's exactly what seems to be the case here in this particular instance. So to me, that really tips the scale, I think, very strongly in favor of this idea of common design, not common descent. And in fact, you could argue, if you look at this phenomena called convergence, which refers to independent evolutionary origin events that produce identical outcomes, but you look at convergence from a design framework, well, it's not unusual for designers to use the same designs over and over again to solve problems. And so you could see that this fact that there's an independent origin event further reinforces the notion that this is an archetype that exists in the mind of a creator that is then functionally manifested in two separate instances, in two separate groups of animals, the protostomes and the deuterostomes, to accomplish and achieve the same purpose. Now, something else that's interesting along this line that favors this idea of an archetype and of an archetype that exists in the mind of a creator in the common design interpretation of the data is the fact that we use retroviruses in biotechnology by taking advantage of the properties of retroviruses that make them very useful for things like gene replacement therapy. In the very early days of gene replacement therapy, researchers were looking at using retroviruses as the vehicles to carry genes into cells that have had their genetic material damaged, with the idea of replacing a defective gene with a healthy gene. And what they would do is they would engineer retroviruses or lentiviruses or the adeno association virus so that they wouldn't replicate in the cell. And then they would incorporate the replacement healthy version of a gene into that retroviral genome, and then allow the retrovirus to infect the target cells, and then because retroviruses will engage in the process of reverse transcription, where the DNA is integrated into the genome, you can now introduce a healthy gene into the genome of the host cell and restore the lost function that resulted from a genetic mutation. But the point is that as human designers, we're utilizing the properties of retroviruses to carry out basically gene replacement therapy, gene editing, because we're taking advantage of properties that these viruses have that we deem to be useful. So if that's the case, and given that we're made in God's image and that we are many creators, why wouldn't we expect the creator to employ a same kind of approach And you could argue that this is what we're actually seeing in these two studies that were published in Cell by these two separate research teams. Something else that I think is interesting to note, and that is so many people look at endogenous retroviruses as being essentially junk DNA that arose as a result of a retroviral infection. And they look at shared endogenous retroviral sequences, for example, in the human genome and the chimp genome, as reflecting, again, a shared evolutionary ancestry. But what this study is indicating to us is that endogenous retroviruses have very useful properties, and could it be that the properties that we're seeing with respect to endogenous retroviruses that are shared among humans and chimps reflect function and, again, common design as opposed to common descent? And it's very interesting because there are people that have noted that when a retroviral infection takes place, endogenous retroviruses will be transcribed at very high levels in genomes, and that transcription actually creates defective protein capsids and retroviral-like messenger RNA that will inhibit the assembly of retroviral particles. That is, it disrupts the infection cycle. Other people have argued that the protein called syncytin, which is encoded by the syncytin gene, actually, again, looks like it's retroviral in nature. Well, some of the retroviral proteins that are part of the capsid are fusogenic, meaning they promote the fusion of membranes with one another, which is part of that infectious process. Well, it turns out syncytin is involved in causing membrane fusion events that are necessary in the trophoblast to form the placenta. And it looks as if, again, from an evolutionary standpoint, that this retroviral gene was co-opted to form the syncytin protein that now plays a critical and central role in placental formation. Now, again, to me, this is another example of common design, not common descent. And I see this as problematic given how central the formation of the placenta is for mammalian reproduction. But on top of that, from an evolutionary perspective, it looks as if this co-option of this endogenous retroviral gene to form syncytin happened 10 separate times in mammals. Another example of convergence, which again doesn't make sense in an evolutionary framework. So when we put this all together as a package, I think what we see here is that if you relax the requirements of methodological naturalism, you have the possibility of presenting a intelligent design / creation model perspective on these results and results like these that make perfect sense. That a creation model or an intelligent design approach to biology readily accommodates these discoveries. In fact, actually predicts these discoveries if you employ this notion of an archetypical design. And in fact, I would argue that given, again, the central role that these so-called co-opted retroviral genes play in a number of critical physiological processes that are in and of themselves very complex, really makes it very difficult to think, at least from an intuitive standpoint, that evolution could somehow have co-opted these genes and then integrated them in such a way to create this kind of complex process. But on top of that, in these instances, we see some of the most astounding examples of convergence, which to me is something that the evolutionary paradigm simply does not predict. So I think by relaxing the requirements of methodological naturalism, we're actually moving closer to what the true explanation is for biology and for the design of biology and the designs of and the architecture of genomes, including the human genome. So anyway, I'm going to go ahead and bring this to a close by just simply pointing out that again, if we think about science in the right way, we have very powerful reasons to believe. And I'm going to ask as I close the broadcast, for those of you that are watching, to take a moment and check in the comment section. Let us know who you are and where you're watching from. Also react to the broadcast using the like button. And then finally, let me know what you think about these discoveries and what they mean for the Christian faith or, from your perspective, for the evolutionary paradigm. God bless each and every one of you. And again, remember this, the more that we know about science, the more that we have reasons to believe. Over and out. See you next time.